right. Good morning, everyone. Thank you very much for joining our webinar today. Today, Westburg, in collaboration with Analytik Jena, will be presenting a webinar presenting you with efficient methods for tissue homogenization and nucleic acid extraction. The webinar today will be presented by Dr. Anne Koch. Dr. Anne Koch is an application specialist with Analytik Jena with the liquid handling and automation team, and she holds a PhD in molecular biology from the University of Essen in collaboration with the Memorial Sloan Kettering Cancer Research Center in New York. First of all, Anne, welcome. Thank you very much for taking the time to present today and present some of the applications that Analytik Jena has for the homogenization of tissue as well as for nucleic acid extraction. With that being said, if you have any questions at all during the webinar, please feel free to post those or write those in the Q&A box. You can find that in the top right corner in the menu bar of the Teams application, and we'll be sure to go through any questions that you have at the end of the presentation. Anne, thank you very much again, and please feel free to take off.
Okay, thank you very much for the introduction, and welcome to everyone to today's webinar. Yes, so I'm from Analytik Jena, as you already heard, and today we're going to talk about homogenization and nucleic acid extraction and how to make that as efficient and easy for you as possible. So first, the general workflow to detect nucleic acids starts with a sampling process, of course. Lots of different kinds of organic matter can be used as a source for nucleic acids. First, these samples have to be collected, of course. Then it's very important to homogenize them, so to reduce them in size and also to make sure that the sample is homogeneous throughout. So it has the same composition all over. So then you can take a small part of this homogenate and go into the extraction process to either get your target RNA or target DNA, or in some cases, of course, you can co-extract both of them as well. And then finally, the goal is, of course, to detect this one specific target molecule that you're looking for. And this will be done with a real-time PCR, giving you a fluorescent signal here and even enabling you to quantify how much of your target was in the original sample. So we from Analytik Jena provide some solutions for you to make this process as easy as possible. So first, for the homogenization, we have our SpeedMill Plus, which is a bead mill that can very efficiently homogenize up to 20 samples in parallel. Afterwards, the extraction process can be automated with the InnoPure C16 Touch, which, as the name implies, can process up to 16 samples in parallel of various different sample materials. And then we also offer PCR devices or Q towers. And this will be the topic of a later webinar, but today we are focusing on the preparation for the qPCR reaction. So for the homogenization, as I said before, the objective is, of course, to have a homogeneous sample. So that means if you take one fraction of your sample, this will be representative of the whole sample. This process is used for various analysis methods, not only for the qPCR preparation. So you can use your homogenate also for other analysis further downstream, but of course, in our case, we're going to proceed with a nucleic acid extraction after the homogenization. And of course, if you want to make it as streamlined and efficient as possible, you want to process multiple samples in parallel. And that's what our devices enable you to do to really speed up the process compared to a manual homogenization or extraction process. So the speed mill, as I mentioned before, is a bead mill. That means we are using these tubes that contain different beads, so they can have different sizes and be made up of different materials. So you can choose the perfect fit for your sample material. Then there are pre-programmed protocols with the speed mill, so you can start the process very fast. But you can also, of course, create your own protocol to really optimize the homogenization for your sample material. And of course, when you have beats at a high frequency pounding on your sample, there will be a rise in temperature. To counteract that, of course, there's a cooling option integrated here. The passive cooling makes it really easy to handle. And also the footprint of the device is very, very small due to that. Um, the lysis tubes can be basically, or we have some requirements as to what the tubes should look like. They should be screw capped and freestanding. I mean, they're going to be hanging in the speed mill, but the freestanding part is important because there will be lots of pressure exerted also on the bottom of the tube. So a V shaped tube would not withstand this pressure. So you need this little fringe along the edge. And we are working together with the IST InnoScreen, which provide lysis tubes of various sizes. So you can here see some samples of those. So they are lysis tubes with 0.5 ml volume and 2 ml volume, depending on your samples. Of course, you can choose whatever is best. Also, the IST InnoScreen provides on their website a very comprehensive overview of which sample materials should be processed with which lysis tube. You can also see the beads are of different sizes here. So you have bigger steel beads, but they're small ceramic beads. And here, this isn't empty, but they are very, very small glass beads as well. So depending on the sample material, you can choose the perfect tube here. And those have been tested extensively with our SpeedMill Plus, so you can really rely on the results here. And then you can proceed to the nucleic acid extraction. Of course, the objective here is to get as much of the nucleic acids out of the sample material as possible. So this can be either DNA or RNA, or you can co-extract both, as I mentioned before. And of course, a very important part, especially for the downstream PCR application, is to remove any inhibitory substances. So depending on what samples you're working with, there may be quite a few compounds in there as well that would inhibit the enzymes of the PCR reaction. And of course, you want to have as clean nucleic acids as possible. And of course, reproducibility is always an issue as well. And again, multiple samples to really streamline the process. So the processing of multiple samples is very advantageous as well. So of course you can do all of this manually, usually, the nucleic acid extraction follows these 5 steps. The cells have to be lysed to open them up. Then the nucleic acids can bind to a matrix. This matrix must then be separated from all the other components of the lysate. And then while they are bound, the nucleic acids can be washed to get rid of those inhibitory substances. And then again, of course, you want to release them from this matrix again, so you can work with them downstream. This can be a very laborious process if done manually, but the InnoPure can actually take all of those steps off your hands. The lysis sometimes has to be done externally if you have very complex sample materials, but there are lots of kits available also where even the lysis is done within the device. So this can save you a lot of time and effort. And while the device is processing your up to 16 samples, you can go and do something else, of course. The InnoPure has quite a few nice features that I want to point out just to show what the benefits are for you if you're using this device. So you're very flexible with the amount of samples, so you don't have to process 16, of course, in parallel, depending on how high your demand is. Here you can choose any number between 1 and 16 to process with the device. Also, you're flexible with the elution volume. You can choose between 20 and 500 microliter. Of course, again, very much dependent on your sample material. When you have very high DNA content, of course, you want to use a larger elution volume so that you can really efficiently wash it off the matrix. And when you have rather low DNA content, of course, then you want to have higher concentrations by just using small amounts of elution volume. Of course, we have the front doors included here. They're see-through, so you can actually see what the device is doing, but your samples are safe from contamination. And also, if you're working with infectious samples, for example, you are also safe. And between runs, you can, of course, also decontaminate the interior of the device by purchasing an optional UV lamp that will decontaminate the whole interior. The process works very efficiently because we have this heating magnet unit incorporated into the device. So the magnets, of course, are needed when you're working with kits that rely on magnetic beads. And also the heating function included here will efficiently or make the lysis and drying steps very efficient, of course. And we've even found that the elution from the matrix works much better and you get much higher yield if you also heat up your eluate a little bit. And the whole device is controlled via this nice big touch screen up here. I will go into the software a little bit later again, but you can already see it's very clean surface. You don't have a lot of buttons. There's not much to do wrong. It's very intuitive, and still it includes everything you may want. So you can document all of your runs. Of course, you can create templates for your various kits you may be using. You will have the project files later on. Of course, there are also some tools included for maintenance, and you can transfer your data, of course, very efficiently as well. The tablet also comes with 2 USB ports on the side, so you can also attach a barcode reader, for example, to make it more easy to input your sample names, for example, if you're working with barcoded samples, or also the kit information. You don't have to input it manually. You could just scan it off of the kit packaging. And to use the InnoPure, basically you only need to do very simply 3 steps to prepare everything. So you can get pre-filled reagent plastics. Of course, these have to be opened and placed in the so-called sample tray here. Here you can also see the 2 different formats that are provided. So you can use a plate, which holds up to 8 samples. And here you can also use separate stripes. If you work with very low sample numbers, you can put up to 4 of those stripes into this adapter to process single samples or just 2 or 3 at a time. And this block up here holds the pipette tips that the device will use, and also the elution tubes. All of these consumable materials are included with the kits that are used by the InnoPure, so you don't have to worry about any shortage of any consumables, because it will always, all of it will be in your kit, and you will not run out. It's always optimized to the sample number that the kit's specified for. And the last thing you have to do is, of course, transfer your samples to the plate so that the InnoPure can do the extraction process. And I've included 2 short videos here just to show you how you can set up. Oh, sorry. Just a question. Did you hear the sound from the video? I'm not sure if you can. Otherwise, I would just talk over the video. Can I get some feedback?
No, we cannot hear it.
Okay, then I will just talk over the video. So you can here see a non-filled plate. So this is also possible. Then you're very flexible, and also the kits have a longer shelf life if you buy them non-filled. So then you will be provided with the bottles and empty plates. This is of course also an option, especially yeah, to prolong the shelf life of the kits. And here this video is just to show that it's actually quite quick and easy to also pre-fill those. But of course, all the kits are also available already pre-filled. You can see there pipette tips for the stepper that I will be using here. And what we are doing is that we reuse the pipettes for the same buffers over and over as well. Of course, you have to change them every now and then, but since you're using the same buffer, as long as you use the same lot, it's fine to use the same pipette there as well, just to also not create so much waste in the lab. So now, I will show in this video how to pre-fill it. So I'm yeah, just again showing the bottles. Those are coming with the kit, of course. Um, and then you can simply decide how many samples do you want to process with this non-filled plate. You're very flexible, of course, with your sample number. If you only have 3 samples, you will only pre-fill the plate for 3 samples, and then you can reuse it later to process the other on the other 5 slots on the plate. And the kit will also give you all the instructions of how much volume of each buffer goes into which cavity on the plate. And then, like you said, saw just filling one row, it's very quick. And of course, you don't have to pre-fill the whole plate as well. So some rows will be left empty because they are needed as waste positions during the extraction process. But yeah, so this whole process only takes about 5 minutes to pre-fill the plate for 8 samples. So it's really very quick. Now, this is showing you how the sample tray is loaded. So the sample tray here is in the so-called priming station, which makes it easier. Like if you don't have so much space in the lab where your InnoPure is, you can also prepare the plate somewhere else and just conveniently carry up to 2 trays at a time with the priming station. And also it holds it as at a nice working height as well. And here I'm showing how to put in the plates and the pipette tips and elution tubes into the sample tray. And like I said before, those are basically the only steps you have to do before starting the automated extraction. There are those clamps holding down the plates so they cannot move, and then they're at the perfect height for the magnet to attach to the bottom of the plate. And in this case, a dummy plate is used on the other side. So you can only work with one plate at a time if you want, but to make sure that the device works properly, a dummy plate is recommended on the other side. And then here, those are the tubes that are coming with the kits. Of course, you may also use your own elution tubes. You just have to make sure that they're not much higher than the pipette tips there, because then the device cannot detect the height of the elution tubes. So just to make sure that the tips will not crash into anything, they have to be rather low. But apart from that, you're also free to choose yeah, different tubes if those do not suit your needs, for example. And then basically the tray is prepared and you can input it into the device. You will start it, and this would be the home screen. And here you can see the surface. I mentioned it before. We only have those 4 buttons, one for the data management. Here you can find all the log files, also the templates and project files are saved here. In the tools section or in the options section, you can find some tools, but also information on the firmware and software, and also update possibilities. And of course, also how to contact us in case you have any questions. Then in the project tab here, you can find the documentation of the previous runs. And in templates, of course, you can create or just load an existing template. So you create a new one or load an existing template. And to set up a template is actually also very easy. We have those 5 tabs over here. This first one would just be an optional input. This is what you are seeing here. So you can give a title to your template, and you can also say who's processing it, but you don't have to. This is for your documentation purposes. The date and time will be given by the device, so you cannot change this. So you can be sure that it's always documented properly, and you cannot alter these preset values. Then, of course, in the kit information, it's mandatory to choose what kind of extraction technology you're working with. I will come to this in a bit again and explain a little bit more what those 2 technologies are. And of course, then you have to choose the kit that you're working with so that the device knows which protocols to load for you, so that you can process the samples most efficiently with the provided kit. And in the extraction tab, those are not mandatory input, but of course it's recommended to choose your desired elution volume. Preset is only the lowest elution volume of 20 microliter, which is actually not advisable in most cases. So you can, of course, increase this. And then optionally, you can also choose different protocols. So for some of the kits, we're, for example, providing fast, standard, or sensitive protocols, depending on if it's time critical or if you want to get as much as possible out of it. The default, of course, is always the standard protocol. Then again, the sample layout is an optional input, so you can also create your own sample layout in Excel, for example, and just upload it here into the device. But of course, you can also create a sample layout within the software so that you know which sample is where. So this again is optional just because it's for your documentation. The device itself will run through its protocol no matter what. So the sample input here does not change the working of the device. And then the monitoring tab is basically just giving you an overview so you can check that all your inputs are correct, and then you can simply start the extraction process. So just to show you what this looks like with the kit information here that you have to, so this is the only mandatory input you have to do so the device knows which kit you're working with. You can choose here between smart extraction and magnetic bead extraction kits. And depending on the choice you make here, you will get a list here in this drop-down menu of the various different kits that are available for the InnoPure C16. You can input all of this manually, or as I mentioned before, you can also get a barcode reader and just simply scan the code on the box. Then all of this information down here will be filled in automatically. So also expiry date and lot number. You can leave those fields empty, but of course it's recommended to also, for documentation purposes, put this in here. If the expiration date is already exceeded, then the software will give you a warning and letting you know, please be careful, your kit has expired, but you can of course proceed at your own discretion. So then it depends on your judgment if you want to proceed with an expired kit, but the software, of course, will let you know. Yeah, and then the next input that you can do but don't have to, but of course it makes a lot of sense to put in an elution volume. So you can do this via the slider here. You can just move this around, or you can put it in by typing the number here. So in 10 microliter steps, you can go all the way from 20 to 500 microliters. As for the protocols, so in the case of this kit here, the virus kit, there are quite a few protocols available. This is not the case for all the kits, so it of course depends on the sample material whether this makes sense. But in this case, there are also fast and even ultra fast protocols available for the 200 microliter sample volume. And you can also put in more sample if you want to have even higher yield in the end. And of course, you have to choose the corresponding protocol so that the device can process more of your sample. Yeah, and then finally in the monitoring, you can just see if you put in a title in the general tab, you can see which kit was chosen and which protocol, and also which illusion volume. Now you could also go to this button down here to save this template. So if you are working with only 1 or 2 different kinds of kits, of course, it makes a lot of sense to just do the general presets once and then just save the template. Next time you can just open up the template and start immediately, just adapting the sample layout as needed. And after that, you can just press the play button down here. It's a bit small, but since there's not, it's a very clean software interface, so you can find it, of course. And of course, you always have a home button down here to return to the main menu as well. And this help button will always take you to the manual of the device and exactly to the page that is relevant to the screen that you're on. So yeah, you're never left alone here. And if anything should not be as intuitive as we've intended, then of course you can always read up on how you can work with the device at this particular step. And then the extraction will start. And I already mentioned the 5 steps before that are usually part of an extraction process. And as I mentioned before, the InnoPure can work with 2 different extraction technologies. So up here, you can see extraction with magnetic particle-based kits. So here, magnetic particles are coated with a silica coating, which can bind nucleic acids. A lot of automated kits work with such magnetic beads. So after the nucleic acids have been bound to the silica surface here, a magnet can collect the beads at the bottom, and you can simply remove, or the InnoPure, of course, will do this for you, can remove the lysates. And since the magnets are ring-shaped, the particles are collected like in a circle around the perimeter of the cavity here. So the pipette tip can actually go down all the way to take out all of the lysate here. So you have very little death volume and also little carryover of magnetic beads since they're on the outer perimeter of the cavity, whereas the pipetting happens in the middle, of course. So this also increases the yield and also the purity of the sample in the end. Yeah, then the washing steps are next. So while the nucleic acids are bound to the beads, there are various washing steps with different buffers depending on the kits. Those are optimized to the material you want to extract. And finally, an illusion buffer washes off the nucleic acids from the beads, and they are transferred to the illusion tubes that you put in the sample tray before. The smart extraction here works similarly, but it doesn't require the magnet. So the extraction here takes place within the pipette tip. So there are some granulates in here with smart modified surfaces that can bind very high amounts of DNA, and also the opening of the tips here is wider than from regular tips, so you have less shear forces acting on the DNA, so you can extract very high molecular weight DNA with those extraction kits. And since the nucleic acids will be located within the tip the whole time, in this case, not the liquids are transferred, but the tip itself moves across the plate to the different buffers that were pre-filled in the plate. So after it's taken out of the lysate, it goes to the different wash buffers. And in the end, of course, it goes to the illusion buffer. And then we're switching to a different pipette tip to transfer the DNA from the plate where the extraction process took place to the illusion tubes in the sample tray. And then the device will stay closed. So the doors will stay closed until you confirm that the end message basically. So if the pure is done, your samples are safe within the closed compartment of the device, and then you can simply take them out and proceed either to store the nucleic acids, of course, or to go to the analysis. So, and now to touch on the different kits that are available. So also like with the lysis tubes, we're working with the IST InnoScreen. Here they've developed their kits especially for the InnoPure C16. All of these kits have been tested extensively with our device and the different sample materials, of course, that the kits were developed for. For smart extraction, for the high molecular weight DNA extraction, there are 3 kits available, 2 of them for extraction from blood and 1 from tissue samples. And the magnetic bead-based extraction kits have an even higher variety. So you can see there lots of very specialized kits depending on what kind of sample material you're working with. So here the buffers and also the protocols like the lysis steps and how many washes and everything that's happening within the device has been optimized for all of those various sample materials. And some of these kits are also available as IVD kits, and then they're called Delta Prep. So this basically concludes my talk here about the extraction and homogenization. We just, as a quick summary to the InnoPure C16 Touch. So we have several decades of liquid handling experience here at Analytik Jena. So of course, we can make sure that everything has been optimized. So there's lots of years of experience going into the development and building up those devices. And we try to make, our main goal is to make you, the customer, as flexible as possible. So we don't want to limit you in the sample number, for example, you want to process, or also the sample types. So we are trying to offer as much variation as possible. And of course, we're also open to new developments to accommodate any needs that may arise on your end. And we hope that our devices can meet your expectations, and we're always striving to get better there as well. And again, of course, we want to make it convenient. So reducing hands-on time, of course, is the main objective when automating a procedure. And so the InnoPure is taking a lot of time off of your hands here. So with this, I want to thank you for your attention. And if there are any questions, I'm very, very happy to answer them.
All right. Thank you very much, Anne, for this very informative and broad talk about the products that Analytik Jena offers that help you to make your tissue homogenization and nucleic acid extraction much more efficient. To everyone at home or in the office watching, I would like to also thank you for your attendance and for your attention. If you have any questions at all in relation to the products or the presentation that we presented here to you today, please feel free to reach out to us at Westburg or to Analytik Jena at any time. We are more than happy to assist you with any questions you have and to help you discover how the Analytik Jena products can make your life and your workflow much more efficient.